Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today we are going to start a new session or a new series and its name is Learn Bioinformatics from the Scratch. In the last few days I received some comments. Also, in daily life, personally, I met some people and they were really interested to learn bioinformatics from the scratch. So I thought, why not to start a kind of series and uh, start teaching it from the very basic and proceed it continuously or gradually to the advanced level. So let's see what we are going to learn today. This slide contains aim of this session. Today is part 1 and we will have many sessions in the coming days. These sessions are basically designed to assess those who wish to learn bioinformatics from the scratch. It means they are beginners or they do not have a good concept. So they want from somewhere to start easy learning or start foundation learning in the field. This session will focus on two key areas. I will try my best to deliver conceptually, and I will also include some demonstrations for you to perform it on your own. Who are my target audience? If you are a student or a researcher with a background in biology or life sciences, you are a target audience. No prior knowledge of computer programming is required at this stage. We will learn, but gradually. Person should know about, I expect that he or she should know about genes, central dogma, or proteins. Okay, so let's start. I'm giving you the first concept, sequence. What is sequence in bioinformatics? When we when we hear this word sequence, what actually it means? So remember, the sequence means either this or this. This is a nucleotide sequence. If you see a sequence that is composed of A, T, C, G or A, U, C, G kind of characters or alphabets, then you can say this is a nucleotide sequence. Another sequence in bioinformatics stands for amino acid sequence, and it will contain some more letters apart from ATCG or AUCG. Apart from these four letters, because they are also included in the amino acid sequence. So don't be confused. Apart from these four letters, they will contains V. You can see V is not here. P, S, T, H, E, Q, N. And technically, we have read in our course books that there are 20 amino acids which codes for proteins or for enzymes. So if you see, if you want to understand which one is nucleotide sequence or which one is an amino acid sequence, so just look into the sequence. If it contains ATCG or the combination of ATCG, it is a nucleotide sequence. Otherwise, it will be an amino acid sequence. So I hope you understand. This is your initial concept that if you are asked to provide a sequence or to use a sequence, then you should know which sequence the person is asking for nucleotide sequence or for amino acid sequence. Now, here is some concept, I name it concept 2. What is the importance of sequence. So let's start from here. Nucleotide ATCG, where they are found, you will find the nucleotide, these kind of characters on a DNA or on RNA. Therefore, I mentioned that ATCG makes DNA or RNA. It means all the DNA or RNAs. RNA include all the other forms like ribosomal transfer and messenger, and if you have any other RNA in mind. So all the RNAs, they are composed of ATCG, but in RNA, the T is normally represented by U. So AUCG, or if it is ATCG, then if you can see T, then it's a DNA sequence. Okay. Remember, okay, just imagine a DNA and a double-stranded DNA, and the DNA contains these sequences. Actually, the DNA is made of these sequences, nothing else. Okay. There are two other things, phosphate and ribose sugar pentose, but we are not going to structure up DNA. So for you, it is important to understand that the DNA is composed of ATCG. Okay. Now, the DNA, this longer stretch of DNA which you have assumed in your mind, let's say it's this one. This is one strand of the DNA. Remember, this strand will contain two parts, broadly speaking. It will contain two parts. Non-coding, we also call it intron, and coding, we also call exon. What it means from the intron and exon. Let's suppose this is the DNA strand you assumed in your mind. You can see there are different colors: red, yellow, cyan, or light blue, and green. So assume that these red are non-coding or intron, and these other colors are coding, exon. But the sequence is present in uniform, just like starting from here and coming up to TAT here. This is kind of an arbitrary sequence I used. Okay, so these non-coding part will not code to some product. What I mean, these products, but to learn about this product, let's comprehend this figure. These red color, I told you, they are non-coding. So we can also say that in this DNA, we have three genes. How many genes? Three. Why we excluded these two areas? Because they are non-coding. So typically, genes codes for some product. So that part of DNA which codes for some product is a gene, and that which do not code is not a gene. This is the most simplest way I can explain to you. Okay. So now, as for this figure, this DNA strand contains three genes: gene 1, gene 2, and gene 3. Now we talked about some products. Remember these genes, because they are coding, they are exon, and these red colors are introns, non-coding. These exon will code for some products. What are the products? The products are actually the amino acid sequences. Okay, so technically this sequence codes this one. Now gene codes amino acid sequence. So it means that for these three genes, theoretically, we will have three products. product of gene 1, product of gene 2, and product of gene 3. Amino acid sequences, then, once they are generated or once they are produced in the cytoplasm, then they fold and form a complete protein. Remember this word. You cannot say this linear sequence produced or this kind of sequence produced at the end or at the end of the translation is a polypeptide sequence. That is not a protein. It becomes a protein or they become protein when they fold. So when they get folded, in that case we call it proteins, because when we say protein, it means the protein should be able to perform functions, and the amino acid sequence individually cannot perform functions. It has to fold to perform function. Okay. Then once the amino acid sequence is produced, it is folded, and in your theory you might have studied the primary sequence of protein, the secondary structure of protein, or the primary structure of primary sequence of protein, secondary, tertiary, and quaternary. So for some protein, the tertiary structure is the final level, where for others our quaternary structure is the final level. So proteins are structural and functional units. You suppose, like these genes, okay, one of them, let's say gene 2. This is the gene 2 sequence. It produced an amino acid sequence. The amino acid sequence folds into a protein. And remember, generally, we are not going so deep into the biology. So, generally, proteins are of two types based on functions. They perform structural functions, or we call it structural proteins and functional proteins. Structural proteins are those which forms the structures of your organs or organelles. Okay, let's say here's nail. So there are proteins. So those proteins are actually called structural proteins. But there are other proteins which are not part of the structures or organs or organelles. They are performing, or they are taking place, or they are playing a role in functions, in different functions which are taking place inside the body. So the functional proteins normally, we call it enzymes. If you pick any book or any website regarding enzyme, so in the first paragraph, you will read this word that enzymes are proteins in nature. So that means that enzymes are that class of proteins which are functional. But why we differentiate enzyme and proteins, or we mark them independently? Because in proteins there are structural proteins also, which are not taking place directly in any biological process. So that's why we cannot say them enzymes. So enzymes are that subset of proteins which are taking place in some biological processes. So once we have enzymes, then we have protein. We actually, that is a proteins, and that will have some sequence. So up until now, I hope I have delivered two concepts. What are sequences in bioinformatics and also in biology, and how these two sequences, these two kinds of sequences are related to each other. Also, they are different from each other. They are related to each other because genes produce proteins or enzymes. They are different from each other because protein sequence is only give you the hints or the information that how it will fold, or how it will make a 3D structure, okay, or a folded structure. But the gene sequence itself is enough. Okay. So once you understand this kind of thing, then it will be easier for us when we will study the sequence manipulations, mutations, and all those things. So that's why I thought that these two concepts are necessary for any beginner. Okay. Now, the next thing is you understand what are sequences. Now, in bioinformatics, you should know how to retrieve those sequences. Okay, we are not talking about structures, so be stay with me. I'm talking about sequences. So if now you should know, or you know now, like we have two kinds of sequences: nucleotide or genes, or the second level is protein, amino acid, or enzyme sequences. Remember, there are wealth of databases, too many databases, but I will teach you from the start because as a teacher, this is my duty to first keep your foot out from somewhere, pick your foot out from somewhere, and keep it somewhere on the landing pad. So, the landing pad for you is to retrieve these sequences. We will use NCBI for nucleotide sequences, or we will use UniProt for amino acid sequences. Now, let's perform some demonstration. So, first of all, I want to introduce you how to retrieve the gene or nucleotide sequences. As I told you in the slides that we will use NCBI. So you should know the NCBI, but if you don't know, let's say I don't know. So I just ask Google NCBI. It will give me this National Center for Biotechnology information. Click here. Once you click here, you will have the official page of the NCBI. Okay, this is the official page of the NCBI. Remember, it is kind of a separate world. Okay, too many informations or too many things are here, but we will learn them gradually. Okay, first of all, I want to teach you how to extract the gene sequence from the NCBI. Just click here, all databases. Ignore all these other stuffs right now. Okay, and scroll down, you will see a word nucleotide here. Click here. Now, write the name of your sequence or a gene sequence. Let's say I want to write this name NDM-1. Okay. In your case, you should know a gene name. If you don't know from the literature or from anywhere, you don't know the gene name, then you may have some information. So write the keywords here, and the internet will help you to find out your exact gene. Okay, click search. Okay, this is our result page. You can see almost 86,807 items. Okay, but on this page, each one is item. So it means right now for this NDM1 sequence, NCBI contains 86,807 sequences. Okay. You can also look to these filters like sequence type. All of them are the nucleotide sequences. Okay. How many of them are included in the reference sequence? Just you can use them as a reference. So this number, sequence length, blah, blah, blah. Okay. Right now, just ignore it. Okay. Now, let's say this is your sequence. Okay, from here it gives you some information. You can read them, like the sequence, this gene sequence is composed of 7 to 9 base pairs, and it is a DNA sequence. And if you want to look out the protein, but don't go there right now. Okay, you can also click FASTA directly. Otherwise, I will not recommend, just click here. Okay, when you click there, you will receive this kind of window, possibly also some information here, but it's okay if it not giving you any further information, that doesn't matter. Okay, look here, complete CDS. CDS stands for coding sequences. Okay, but I know I already gave you the concept too, so you will understand what does it mean. So this is the indium 1 gene, and all the sequences which codes for something, for some protein, for some product, the status of this record is complete. So most of the time students, they don't look here and they just pick one sequence, and later on they come to know that, oh, we selected a partial sequence. So actually that was your mistake, you didn't read at the start. Okay, now click here, FASTA. Let me refresh. Okay, when I refresh, I got the full page. Okay, so now this just we will take 1 minute or less than 1 minute to understand this data. This gives you the locus, total length. It's a DNA, it's linear, it's from bacteria, it was submitted on this date. This is a from which bacteria? Klebsiella pneumoniae. This is the accession number. Keywords you may see for some, may not see for other, doesn't matter. This is the organism from which the gene sequence belongs. It is the complete classification. And who submitted this structure? These are the names, these are the title of the journal is still unpublished. That says, okay, and the sequence submitted from India, AMU Aligarh, and assembly, which technology they use for solving this sequence. So they use the Sanger technology. Okay, maybe this is not important for you, but it may be important for someone. Okay, this is actually the gene sequence, all 729 nucleotides. Okay, now click here, FASTA. Once you click here, the FASTA file, now you will be able to see this. Just copy this, let's say copy, and do not copy them into your Word file, just new, and select the text document. Most of the people, they are copying these elements or these things in the Word format, so that's the wrong way. I will name it NDM-1 and Control V. So this is technically your sequence. We retrieved a sequence of first gene if you are new in bioinformatics, and we saved it on our desktop. Now we are going to access another sequence, another database. So if you don't know, just write UniProt, UniProt, and universal protein. That stands for. Okay. Okay. So this is the UniProt, and you you need to type any name. Okay. I will type the same name NDM1 again. Search. Okay. These are all the entries. If you see it says 91,056 result for the indium one. You have to write your protein name there and it will pick it. The second thing you should care about, from which organism you are interested. All the organisms are listed here in the organism tab. Okay, this is a protein name. Okay, now one thing more I want to discuss. You can see three golden spots, colors, but for the other sequence, this area is not golden. Why? The golden means reviewed sequence. It means these three sequences among these 91,056, these are manually reviewed by the Swiss Prot administration, and you it's safe to use. Otherwise, you can also use the other sequences. That doesn't mean that they are wrong, but they are not reviewed. Someday they will also be reviewed. So that's okay. Let's say I'm going to open this. Okay, this is our first the first this is the entry I clicked, and it tells you the gene name and it tells you the protein name. Okay, if you want to read, I suggest you to read, but if you don't want, our job is to learn how to extract the sequence, so just click here sequence. Now once you click here sequence, copy here, copy sequence. Now we are going to save this sequence back in the same window here. Um, in this text file, we already saved this sequence from our nucleotide search. Okay. I also want to tell you one thing more. This sign, this one, we call it header in bioinformatics. In mathematics, they call it greater than. So if you missed a greater than sign for a sequence, let's say you paste this sequence this way, no sign, then the bioinformatics programs will not read this sequence. So that's why, remember, for every sequence, the first mark will be this one. We call it header, and once there is a header, so the programs will then followed by a name. Let's say I name it indium1, enter, and then I paste the sequence copied from the UniProt. Now, if we export or if we open this file in any program we will do in future, then programs will understand that this data contains two sequence records, one, and this is sequence record two. But if you miss this thing, program will not understand the second sequence. So it was the importance of the header. Okay, for today, I think it is enough. My main goal was to deliver you two two concepts and to teach you how to extract sequences if you are a beginner. And in next sessions, we will carry on and we will proceed gradually to different advanced levels. Thank you so much. If you are liking these things, please share with your juniors or with your other friends. And thank you so much. Bye bye.